My name is Eric McDonald. I'm a Plant Protection Quarantine Officer for the United States Department of Agriculture. I work at the Plant Inspection Station in Humble, Texas, and I'm an area identifier with a specialty in acarology. Today's presentation is an introduction to the mite family Tetranychidae. The Tetranychidae are commonly known as the spider mites, and it is a large group with worldwide distribution. All spider mites are phytophagous plant feeders, and several are major pests of many crops, including apples, cassava, citrus, cotton, roses, and other ornamentals. The family Tetranychidae is the most important member of the Tetranychidae. The spider mites are very important quarantine pests, and their feeding causes large economic yield losses to ornamental and agricultural plants. Spider mites are round in shape. Average size is 0.5 millimeters long, or 1/50th of an inch long, and vary in color. Most common color of the mite is translucent, with variations of brown to orange-red or green to greenish yellow. And the family is made up of two subfamilies consisting of 71 genera, with over 1,200 species described worldwide. Spider mite development differs between species, but in general they complete in a life cycle under optimum conditions in 5 to 20 days. The length of time from egg to adult varies greatly depending on temperature. The life cycle is composed of the egg, the larvae, two nymphal stages, a protonymph and a deutonymph, and the adult. The females lay small, whitish, transparent spherical eggs with a diameter of 0.1 to 0.15 millimeters. Most spider mites spin fine strands of webbing on the host plant to help protect the colony from predators, and that is how they get the spider part of their common name. The eggs are attached to these fine silk webbings in the groups that do spin webs and hatch in approximately 3 days. Eggs are laid one at a time, with females depositing 5 to 6 eggs per day, with a total of 60 to 100 eggs per female. And eggs hatch in 3 to 6 days into a first instar larvae. It is colorless when it hatches. It turns yellowish or pinkish after feeding and has 3 pair of legs. There are 2 nymphal instars after the larvae, called the protonymph and the deutonymph. These both have 4 pair of legs, as does the adult. Larvae and nymphs complete development in 4 to 9 days, and there are many overlapping generations per year. The adult female lives 2 to 4 weeks and is capable of laying several hundred eggs during her lifetime. Adult females are slightly larger than males, oval in shape, and outnumber the males. And sometimes you'll find populations on a host where that's all you'll find is the females. Matter of fact, it's kind of discouraging a lot of times, because when we do get interceptions to do identification on, all we have is females to do that with. And a lot of times you need the male to get to species ID. The female has an elliptical body that bears many pairs of dorsal setae. The male is elliptical in shape with caudal end tapering and smaller than the female. Adults live about 30 days, although color and number of spots is frequently used for rough identification in the field. Accurate identification requires microscopic evaluation by an expert. Females overwintering may occur on many wheat hosts in warmer climates, but females may also overwinter in debris in a state of diapause. Adult females are commonly present, as I said earlier, while males are usually rare. And that's why you need to try to collect as much as you can. And when examining a slide-mounted spider mite, the females are easy to distinguish by checking for the presence in the genital region here, and you'll see this like crinkly striations in the genital aperture here, and that definitely is a female. The male is elliptical with caudal end tapering and smaller than the female. And the axis of knob of the male structure here, called the adiagus, is parallel or forming a small angle with axis of the shaft. It is necessary to slide mount males laterally to be able to see the adiagus. The shape of the male adiagus is required for species level identification. All mites have needle-like piercing sucking mouth parts. They attack a wide host range of plants feeding on the leaves, stems, and fruits. Spider mites feed by penetrating the plant tissue with their mouth parts and are found primarily on the underside of the leaf. The mites generally feed and reproduce on the lower leaf surface, but when populations are high, they will readily infest the upper leaf surface. Spider mites are stationary when feeding, but when disturbed, will walk fairly quickly. Symptoms of mite damage include the presence of fine chloritic flecking and presence of fine webbing in those that spin webs. And the feeding damage causes yellowing to bronzing and defoliation. When collecting mites from plants, it is useful to collect all different stages and sexes and note the mite color, feeding damage, and webbing if present. Collecting mites is important for species-level identification of several genera of Tetranychidae. They have an extremely wide host range, damaging many weeds, fruits, ornamentals, woody, and many herbaceous plants. And spider mites prefer hot, dry conditions, so they tend to increase in population during the summer. They attack plants by feeding on the leaves, stems, and fruits. And when spider mites remove the sap, the mesophyll tissue or parenchyma collapses and small chlorotic spot forms on each feeding site. Continued feeding damages the plant. The leaves turn yellow, gray, or bronze. Wilting, leaf deformity, desiccation, and complete defoliation may occur with prolonged high-density infestations. The tetranychids represent a very real threat to our food safety. Potential pathways into the country should be monitored, including passage baggage, commercial maritime and air cargo, military cargo, and land border movement. Improving taxonomic knowledge and developing diagnostic tools to rapidly and accurately identify mite species are critical for the exclusion, elimination, and management of plant-feeding mite pests. The best control program for invasive species is to prevent them from being introduced in the first place. When looking for spider mites, one needs to develop the eye for the tiny. Check for signs and symptoms of mite damage. Inspect the undersides of the leaves closely for mites, cast skins, and webbing. A necessary tool for detection of spider mites is a 10x or 15x magnifying glass. A table magnifier can be useful also for quick scanning of suspect material. Check the stem joints and leaf ribs for mites and eggs. Check plant domatia sites for mites. Plant material can be sampled by placing in a plastic bag or a container and taken back to a facility where you can examine it using one of the following methods. One way is just by putting plant material under a microscope and just checking it out. Also, you can use sieve filters to wash the sample. You wash it under 2 different filters, you collect the rinsate, and you look at that under a microscope, and what you find then you can collect and slide mount. Another way is using a Berlese funnel. You place the sample in the funnel, you turn on your heat source, and you collect samples out of the bottom of the jar. Just make sure to watch it and turn the lamp off, otherwise you might have a fire on your hands. And another method is using seed burrow pans, where you actually shake the plant material in the pans and then dump it onto a white background or a white piece of paper, and check the white piece of paper for any mites that you might see. Look for movement on the paper if you see small specks moving around. If they're moving quite rapidly, it's usually predator mites. If they're moving slower, then you have a good shot there of maybe picking up a plant-feeding mite. The ability to diagnose the following terms and characters in the Tetranychid family will provide an understanding of the taxonomy of the group. It will allow you to recognize and help you to identify spider mites. Let's proceed to check out some of these qualities. The naming of the body CD is called chatotaxy, and it can be confusing in this group due to different seedal nomenclature systems used by different taxonomists. Here are 3 examples of seedal designation systems. And it's the Grangian system using the D, C, the numbers here that were previously discussed. is the most accepted, and that's the one we'll be using. But keep in mind, dependent on the author of references that you might become encountered with, that you may see some of these other systems. And here's an example of the dorsal surface, the epithysome, the protosome, and the Ctul naming. All members of the family Tetranychidae have 5 segmented palps with a thumb claw. The palp also contains a spinneret for producing a web used to protect the mites and eggs from predators in those groups that have that. The gnathosome consists of the chelicerae, palps, and mouth parts, and the palp tarsus has an enlarged spinneret or epithelium in the Tetranychidae, and this is used to spin webbing in many species. Its size and shape is of taxonomic importance. The prodorsum normally has 3 pairs of setae and 2 pairs of eyes. The epithysomal setae can vary from 12 to 10. The Tetranychidae is separated into 2 subfamilies, the Bryobini and the Tetranychidae. Each subfamily consists of 3 tribes. The 2 subfamilies can be separated by examining the impodium and the genital anal setae. Notice the convoluted transverse setae on the ventral side of the genital aperture. That tells you got a female. Bryobini mites have an impodium with tenon hairs, while the Tetranychidae mites, the impodium may be absent or, if present, without tenon hairs. Bryobini female mites with 2-3 pairs of anal setae, and males with 5 pairs of genital anal setae. Tetranychidae females will have 1 or 2 pairs of anal setae, and males with 4 pairs of genital anal setae. The 3 tribes of Bryobini can be separated by examining the impodium and claws. The Bryobina has paired claws uncinate and impodium pad-like. The Hystrocynine has paired claws, impodium pad-like, and while the Petrobini has paired claws pad-like and the impodium uncinate. The 3 tribes of Tetranychidae can be separated by examining the impodium, tarsus leg 1 and 2, and the epithysoma. The Tetranychidae has an impodium claw-like when present, tarsus leg 1 with loosely associated setae, or with 1 pair of duplex setae. If 2 pairs of duplex setae on tarsus leg 1, then no pairs on tarsus leg 2. On tarsus 2. And I know this sounds confusing, but we're going to have, slide samples that we're going to be able to look at, and we'll be able to get this. The Tenopalpidine has impodium claw-like or split distally, ending in a tuft of hairs. Tarsus leg 1 with 2 pairs of duplex setae, and tarsus 2 with 1 pair. The epithysoma with F1 in the marginal position or absent. While the Tetranychidae has impodium claw-like or split distally, ending in tufts of hair. Tarsus leg 1 with 2 pairs of duplex setae, and tarsus 2 with 1 pair. The epithysoma with F1 in normal position. And here's an example of duplex setae, and here's what we're looking at when we're talking about associated setae. Because this family is so large, only 6 genera that are most frequently encountered will be covered in this presentation. They are the genus Tetranychus, Oligonicus, Panonicus, Eotetranychus, Eudotranicus, and Bryobia. The genus Bryobia contains 124 described species, and many of them are economically important pests that are highly polygamous and occur without males. The common name for Bryobia are the clover mites and brown mites. The genus Bryobia contains a number of economically important pests, including several that are not yet reported from the U.S. A well-known and widely distributed pests include the clover mite Bryobia pretiosa, a pest of over 250 kinds of plants. Clover mites are widely distributed in North and South America, Europe, Asia, Africa, and Australia. Clover mites feed on a wide range of plants, including clover, dandelion, lawn grasses, strawberry, shepherd's purse, primrose, and ornamental flowers. They do not cause any apparent harm to turf grass, but their feeding activity can turn the grass a silvery color and may stipple plants when heavy populations are present. Clover mites can become a nuisance in and around homes. Fortunately, when they do come into homes, they do not reproduce and will die within a few days from dehydration. Most of the other Bryobia are important pests, are similar to the clover mite in appearance and are difficult to distinguish. Differentiating the species from other members of the Protosea complex is difficult and best done by specialists. And when examining slide-mounted Bryobia, the claws are hooked. The apodium is pad-like and with more than one pair of tenon hairs. In Bryobia, the size and shape of lobes at the bases of seta V1 and V2 are of importance in species identification, and the number, location, length, and structure of dorsal setae are of taxonomic significance. The epithysome with 12 pairs of setae, and they're typically club-shaped. Bryobia of current quarantine interest include the Bryobia gramenum, Bryobia kissaphylla, Bryobia langodecini, Bryobia vescivi, and this mite sometimes is confused with some of the true spider mites, Tetranychidae. The EO Tetranychidae are important polyphagous pests with 180 species described worldwide. The most common is the six-spotted mite that attacks grapes, cherry, figs, citrus, azalea, rhododendron, figs, strawberries, guava, and a variety of other trees and herbs in much of the world. Many other species occur in the United States, like the yellow mite, the wilmot mite, which attacks grapes, fruit trees, and a variety of ornamentals. The azalea white mite is a pest of azalea and attacks passion fruit, blackberry, and cherry. And the pecan leaf scorch mite attacks pecans, other hickories, and a variety of ornamental trees. 3 species are of concern to U.S. quarantine at the moment. They are Eotetranychus sandani, which is on citrus, muria, legumes, and grass in Southeast Asia and the Philippines. Eotetranychus falcatus, which is on cassava, fig, cotton, jute, peanut, cashew, ornamentals in much of Africa and the Cape Verde Islands. And Eotetranychus canchitis on citrus, grape, rose, and ornamental trees in China, India, and Japan. Key characters to genus are 2 pairs of anal setae, 2 pairs of perianal setae, and CDH1 terminal. Also, the impodium is split distally into 3 pairs of hairs and no spur present. Tarsus 1 duplicate setae is distal and inserted near each other. In addition, the dorsal setae are simple and longer than the distance between assertions, and transverse setae present between setae E1. Panonychus is a small genus of about 15 described species. The common names are the citrus red mite, the European red mite, and the raspberry red mite. They are broadly distributed and highly polyphagous pests. Panonychus are characterized having 2 pairs of perianal setae, 3, 2 pairs of anal setae, impodium claw-like and as long or longer than the proximal ventral hairs, and dorsal setae are usually set on strong tubercles, and paratremes are recurved distally. The European red mite is native to Europe and was first introduced into the Pacific Northwest in the early 1900s. Since then, it has become established throughout the United States and Canada. It is a pest of palm and stone fruits, nuts, berries, and some ornamental trees and shrubs. The citrus red mite is associated wherever citrus is grown, but it also feeds on roses, pears, almond, and some broadleaf ornamentals. It is a serious pest of citrus in Florida and California. Females are red and plump and about 0.5 millimeters long. Males are smaller and taper to the end. The seedy hairs are white with red bases. Tetranychidae are characterized by having two pairs of perianal setae, H2 and H3, two pairs of anal setae. Impodium is claw-like and is long or longer than the proximal ventral hairs. Dorsal setae are usually set on strong tubercles, and paratremes recurve distally. Only one exotic species, Tetranychidae elongatus, does not occur in the U.S. and is of quarantine interest. It attacks beans, citrus, figs, papaya, passion fruit, pear, and ornamentals such as rose in Asia, Australia, and New Guinea. It is widespread in the tropics and subtropics, and it is smaller than citrus red mite and has shorter setae. Other tetranychidae have either one pair of perianal setae, as in like the Oligonychus, Tetranychus, and Aphotetranychus, or have the impodium split distally, ending in a tuft of hairs, or divided into 2-3 claws, or lack proximal ventral hairs, or have hairs longer than the claws. The Tetranychidae are known as true spider mites and are the only genus in the tetranychidae that do not have impodium with tennate hairs. More than 30 species of Tetranychidae have been described, and most feed on a limited number of food and are considered oligophagous, species of little economic interest. The genus is characterized by having two pairs of anal setae, PS1 and 2. The impodium claw is minute, appearing absent, and tarsus 1 lacks two distinct pair of duplex setae, and the opithosoma with 10 pairs of dorsal setae. Except for these three species, which are important pests of citrus and attack a wide variety of other crops, the Texas citrus mite is established throughout the warmer parts of North, South, and Central America, including the USA. In the United States, it occurs in all of the major citrus production areas. It has a fairly large host range, including among others almonds, figs, croton species, and citrus. The African red mite is an important pest of citrus and attacks a wide variety of other crops, including apple, peach, grape, guava, papaya, cotton, eggplant, and a variety of ornamentals. It is widely distributed in Africa, occurs in India, Southeast Asia, Papua New Guinea, and Indian Ocean Islands. The Oriental red mite is probably the most important pest species in this genus with a wide range of hosts. It is an important pest of citrus in many parts of the world, but only a sporadic minor pest in Australia. Hosts include many types of fruit trees like apple, avocado, apricot, peach, plum, also grape, blackberry, bananas, walnut, ornamentals, melon, sugarcane, maize, sorghum, eggplant, pepper, sweet potato, cassava, and beans. It eats about everything. The mite is established in much of Asia, widely distributed in Africa, the Middle East, and eastern Australia. The Oligonychus grass mites come right after the spider mites as far as numbers of agricultural pest species, and they attack a wide range of host plants. So far, about 200 species of Oligonychus have been described, including many prolificous pests of high agricultural and forestry importance. Oligonychus are commonly called the red spider mite, Banks grass mite, southern red mite. They have a lot of common names, like most mites do. A few of the important pest species currently not known to be in the U.S. include Oligonychus afrasiaticus, Oligonychus berrensis, Oligonychus gossypi, Oligonychus indicus, Oligonychus leachi, Oligonychus megiferus, and Oligonychus mcgregori, Oligonychus shinkaji, Oligonychus thelotocus, and Oligonychus zea. And there's more, but that's some of the more important ones. The genus is characterized by a strong sickle-shaped claw like impodium, and with generally long proximal ventral setal hairs on female, and duplex setae on tarsus 1, with one pair of perianal setae, H3, setae H2 terminal, setae H1 absent. with 2 pair of anal setae, PS1 and 2, and with 10 pairs of opisthocidal setae and 3 pairs of protodorsal setae. The last genus is the Tetranychidae genus that contains about 140 species that have been described worldwide, and that list is growing. The most economically important species feed on many host plants, and the more prolific is the pest, the more likely it is to be introduced. This mite has a long list of common names, but most people recognize the 2-spotted mite, the 4-spotted mite, or the carmine mite. Members of the 2-spotted mite complex attack an extraordinary diversity of economically important plants throughout the world. They have been reported to feed on more than 900 different plant species. Tetranychus urtica, the 2-spotted mite, is the most well-known cosmopolitan species in the world. The mite was originally described from European species and is considered to be a temperate zone species, but it also found in the subtropical regions. And it's probably a mite, too, that has been the most misidentified. All species of Tetranychus overwinter as adult females that are colored orange or red, and in tropical and subtropical areas, the mites may remain active year-round. Spider mites move from plant to plant by walking, but the primary means is by wind-borne migration. And here's an example of a larvae. Adult female and an adult. And the following are considered exotic species and pose a potential threat to U.S. agriculture. And I am just going to list these. Diagnostically, the genus Tetranychus is characterized with 1 pair of perianal setae, H3. CDH2 is terminal, not in perianal position. CDH1 is absent. The impodium split into 2-3 pairs of proximal ventral hairs, and the medial dorsal impodium spur generally present, sometimes well developed. Duplex setae of tarsus 1 well separated, and paratremes recurve distally. Simple. And here's the paratremes here. And here's showing the duplex setae and the proximal ventral hairs split and the impodium split. And in summary, spider mites are the most economically important plant feeding species on many commercial host plants. They have a wide host range, are hard to detect, and that makes them more likely to be introduced. Identifying spider mites is tricky due to the fact that males are necessary for species identification, and female populations are frequently encountered. Males are smaller than females and must be slide-mounted laterally to view the shape of the Aedeagus. Species identification invariably require a good lateral mount of the male Aedeagus, and even then, only a specialist with extensive experience is likely to feel confident of the identification. Any questions?
What are some useful diagnostic characters for identifying Tetranychidae and Tetranychus species?
Yeah, there are 3 families in the Tetranychidae that we're interested in, and that is the Tuckerellidae, Tenopalpidae, and the Tetranychidae. And the way you tell the Tetranychidae and separate them out is that they have a very modified thumb claw process that has this claw-like setae at the end of the thumb claw, and it is not linear. And that's how you separate out the Tetranychidae.
How can I differentiate between males and females in order that I can mount males laterally on slides?
Okay, the way you go about differentiating male and females is the females are usually larger. The males will be smaller in size, and the body shape of the males is more oblique and narrower. So when you do slide mount the males, you must slide mount them laterally. And that's why it's important to be able to distinguish between males and females.
What is the name of the current system of ketotaxi that you use most often?
Yeah, it's the Grangian system is what we use when we name the ketotaxi. And we use there's prodorsum cetal names, VI, VE, SCI, and on the epithysome we use C, D, E, F, and H.